Hello everybody. I hope you can all hear me clearly and see the slide. I'm sorry I'm a little sick today, so I apologize if there is any disturbance in the voice. But welcome to the sixth session of the discussions that are held as a part of the NPTEL course Cell Biology Cellular Organization, Division and Processes. So these discussion sessions have basically been like discussing previous assignment questions and discussing the overall concepts of what was taken up in the course that has been instructed by Professor Shrikala Loria from IISc Bangalore. I am one of the PMRF TAs of this course. I'm Smriti from IISc Bangalore. And there are 3 such sessions that happen every week, and you can choose any one depending on your availability. So let's start with this week's session. So this week, basically what was discussed was the packaging of DNA in eukaryotic cell. And we know that this is a very complex process. There is a conundrum in the packaging because the DNA, which is several meters long, if you just pull it apart and arrange it in one linear fashion, that has to fit inside a nucleus that's hardly a few micrometers in length, right? And another fascinating thing about the DNA's packaging is that we all know in eukaryotic cells, this is concentrated within a cell's nucleus. And in bacteria, okay, thank you. In bacteria and prokaryotes also, this is concentrated in a single region. But in eukaryotes, it's enclosed within a membrane-bound space called as the cell nucleus. And this is hardly 10% of the total cell volume. And the cell itself is microscopic in size, and it's not visible by the naked eye. And this entire length of genetic material that makes you is essentially packed inside this tiny, tiny space. So this is just a sequence of a protein coding region taken from Homo sapiens, which is humans protein coding region, and we can see the mere size of it, right? It has so many bases, and they're all just 4 bases. And what is unique to you is the sequence in which these bases are arranged next to each other. So this is what creates any genetic variation, and DNA is nothing but a long sequence of varying order and length. Now, this is how you'll get this DNA sequence from the gene bank that is present online, which acts as a source for all these different regions. And they are all annotated, which means that they have been defined. This region codes for this particular protein, and so on. And typically, the rule that they follow, it's written from the 5' end to the 3' end. So this sequence is arranged from the 5' to 3' end, 5' end being the starting and the 3' end being the end. So every gene we can see here has this unique accession number. This is the DNA, that is the DNA that encodes for the mRNA, and then the mRNA will encode for the protein. So faster sequences are available for DNA, mRNA, and protein. And each of these will have a unique accession number. So if you type in just this accession number, then you will be able to access it directly. So this sequence has been retrieved from NCBI database. So this is the home page of NCBI. This is what will show up if you just type in NCBI into Google and open the first link that shows up. So I'll just briefly go over how you can retrieve these gene information. So here, if you can see, there is a scroll down menu, and there are lots of databases that you can choose from. So because this is a gene, right, we're looking for a protein encoding region, which is known as a gene. We will select the gene database and search for any protein of interest that we want to search for. In this case, I had taken the protein coding region of keratin that makes up our hair, our nails. So here you'll get a list of all the keratin proteins that this database could find, and you can see that it is from different organisms. You can find one from elephant, shark, chicken, Arabian camel. Now, in this case, if you want to get it from Homo sapiens, you can see on the right over here that there's a list of top organisms. If not for this, you can also go to manage filters and select the organisms uh, uh, protein that you want. So I'm just selecting Homo sapiens in this case. And it is like you can see showing a large number of keratin sequences that have been annotated by different authors. So the first one that is displayed over here has an ID of 3875. So again, if you just type in this ID, then you will get the entire information. And here you can see that it is a large page that has loaded, right? Very intimidating page. But the thing is that it contains every information that is required by us. It has this organism here, what its classification is, how this was basically like summarized in the paper, and the uh, like this is the paper that characterized the sequence, and it's just a summary of what keratin is. And then moving on down here, we can see genomic sequence, the chromosome that it is present in, and FASTA. So if you just click on FASTA, you will get the entire coding region present in the DNA. So this is the entire DNA region that has been known to code for keratin. But again, if you want specifically the mRNA region, then you can again scroll further down. And here you can see this, as we already saw, is the genomic sequence, whatever sequence is present in the DNA. Now, a region of this is inscribed into the mRNA, right? And this sequence, typically starting with NM, that is a trick that you can use to distinguish between mRNA and protein. NM is typically mRNA, and NP is typically the protein sequence. So if you click on the mRNA sequence, as in the accession number for mRNA, it will again open a page. And if you click on FASTA over here, you'll get the sequence that encodes for the mRNA. So this is as you can notice, the keratin sequence was longer present in the DNA, and a fragment of it has been transcribed into mRNA. Now again, we go back, and this time, if we click on the NP accession number, then again a similar page is opening up, again having all sorts of information about the authors of the paper and the place wherein this protein was characterized from. And again, clicking on FASTA will give you the amino acid sequence. So here we can see that amino acid, all the 20 amino acids that we know, have been given a single letter code, and this code is how it is used to represent the sequence. So each of these letters stand for one amino acid each, and as you can see, this is much more shorter than the mRNA. So going back to the presentation, how do we know all these sequences? Like, how do we know what region of the chromosome is encoding what, and what is exactly the sequence? This is thanks to a pioneering project that was carried out in the late 1990s and the early 2000s. This is popularly known as the Human Genome Project, and I'm sure all of you would have heard of it. What they essentially did was they took a bunch of organisms that they classified as the model organisms. These included E. coli for the prokaryotes, for plants, Arabidopsis thaliana, for nematodes, C. elegans, and in that order for mammalian species, Homo sapiens, that is a human genome. They collected DNA from various volunteers. It was not just one volunteer's DNA. They collected DNA from various volunteers and then started sequencing them. So in those days, sequencing was also not that advanced like it is today. So it took a lot of time. And imagine sequencing the entire 23 chromosomes one at a time. They used this type of sequencing known as Sanger sequencing that is almost obsolete today, but this is what helped them determine the exact order of bases. So even though it is different from one person to another, we need to appreciate the fact that nearly 99 or more percentage of the genetic material is the same from a human to a human, and that's what essentially makes a human a human. There are certain shared characteristics, and you have to develop a brain or two legs, two arms, and those are includes the majority part of the genetic material, and that is the same, and that was what they characterized. They sequenced. So from there, various other researchers have gone ahead and annotated these sequences. They have given regions. These are gene coding regions. These are promoters. These are enhancers. Different functions have been annotated to these regions, and that is the information that we have today. So how did they go ahead and sequence it? What was Sanger sequencing? So essentially, you have the entire genetic material that needs to be chopped into small fragments of DNA that are workable with, because this will ultimately be run on a gel, and the entire chromosomal length of DNA cannot be run on these electrophoresis gels. So these gels are very much different from the electrophoresis gels that we are used to today. Typically, the ones that are run in the lab today is like this long, right? Maybe 15 centimeter, 20 centimeter long. And these Sanger gels were really like meters of length in order to achieve that proper resolution of bases. So essentially what happens is you take your DNA template, whatever your DNA template fragments are, and then you add primers to it. So these are universal primers, they bind to regions. And then you add the polymerase enzyme, the enzyme that is capable of adding bases based on a template based manner, right? And then what else do you need? You need DNTPs. DNTPs are the bases which these polymerase enzyme adds, and these DNTPs, deoxy DNTPs basically are of four types: ATP, CTP, GTP, and TTP. These are the four bases that are present on your DNA. So essentially, if you just add all of this together, then the primer will bind with the template and get elongated. But the trick is you need to be able to identify what base has been added where. So for that, there is this termination reaction, and there are these dideoxy nucleotides, which basically once they are added, the reaction is stopped there. There is no further addition of nucleotides. So essentially, say there is a mix of all of this and a mix of all of this as well. This much lesser in concentration when compared to normal deoxy DNTPs. There will be random addition of these termination bases, and the reaction will stop there itself. But again, these will not be able to be differentiated, right, from one to another. So what they did was they took four reaction conditions. In all four, there were polymerases, there were templates, primers, and the normal deoxy bases. But it contained only one type of termination basis. So one reaction will have either TTP termination base or CTP or ATP or GTP, just one of them. So in this scenario, what happens is there is normal binding when extension is happening. Say there is a G present at the end of this primer. So the first base that is added will be GTP, and the reaction will stop there also. But there are so many DNA fragments present that in one, if it stops in the first position itself, in another, there will be normal addition of G, and then it will carry on. And then the second region where the G appears will be added with the termination base. And like so and so forth, there will be a large number of lengths of fragments, and they would have all been terminated at regions where that particular base is present. So you take these four reaction conditions and then run them on really long electrophoresis gels, and based on their length, they will vary in their migration. So say in the first condition, where the G termination base was there are only 4 bands seen. But in C, there are more bands seen, and in A, even more bands. So this, based on the length that it has run, if it's run from this direction to this direction, then this is the largest, right? Largest fragment, and this is the smallest fragment. So based on that, you can form the entire sequence of the DNA. So if after primer, this is the shortest fragment, then this means that after the primer, the first base that was added was A. And then the second base that was added was T. So it's one nucleotide more, the molecular weight of this drug. And then the next one was G, then C. So and so forth, you can sequence the entire fragment of DNA that was present in the sample. So nowadays, this has been advanced, that nowadays the DNA sequencing techniques that are used is known as next generation sequencing. Here you do not go through the painstaking way of fragmenting the DNA and then isolating the fragments and taking one fragment and then adding bases to them, running such huge gels and sequencing them one by one. What happens is you extract your DNA and then you fragment them like in Sanger sequencing. But here at the end of it, you add these adapter sequences. These are very essential because this is what helps them attach to these microplate like readers. So there are a lot of DNA fragments of different sequences. In Sanger sequencing, they're all of one sequence only, one DNA fragment at a time. Here, it's not that case. There are all different fragments that are present next to each other. And there is this very powerful microscope. What it is capable of doing, it's capable of exactly taking a picture with every time a new base is added. So in this case, all the different bases are tagged with 4 different fluorophores. So in that plate, if there is in one place, if an adapter sequence is attached, there is only one fragment in that place. So whatever primer based extension is happening over there, it is very specific to that fragment. So likewise, there are hundreds of fragments on a single slide, and the microscope is able to capture at every time a nucleotide is added in all these different fragments at the same time. So this cuts short the time taken for genomic sequencing by a large margin. And at the same time, multiple fragments can be imaged, sequenced, and then later on it is paired onto each other. And these overlapping regions that will be present are overlap, and then the entire sequence of DNA can be sequenced that way by mapping it back. Like if you know a region of the gene of the chromosome, then you place it, okay, this is from this chromosome, and then whatever sequence to the left and right of it you have sequence now, you can arrange accordingly. And there are softwares available for all of these steps, and nothing of this is manual anymore. And you can get DNA sequencing done really, really quickly. So, like we saw in the NCBI database, a chromosome having a DNA encoding region, say this is the entire DNA present that has approximately 40 genes according to this representative. This particular region, you can see that there are only 4 genes, but there are lots of intergenic regions present that are not going to be made into RNA. These have all your promoters, all your enhancer regions, all your transcription factor binding regions, and these repetitive regions that are present in between your coding regions. Now, even if you take this one gene that is made now into an RNA, you can see that it is made up of exons and introns and regulatory DNA sequences. Regulatory DNA sequences again becomes these ribosome binding regions and your initiation regions of initiation of translation regions. And only these exons are going to be made into proteins. All of these introns are going to be spliced out. So this is again not the mature RNA. The mature RNA is much, much smaller than this. And you can see that there is a lot of wastage, right? So essentially to encode the entire protein that is present in your body, this much amount of DNA is not required. It's so much of chromosomes, and in the end, it's only making like say 27,000 proteins of sorts. Then why do we need, like, what is the purpose of these intergenic regions and introns present? So one major function that we can think about is if there are mutations that are going to be randomly inserted into the gene, because their concentration and their volume is so much more than the actual coding region, it's much more likely that the mutations are inserted into this intergenic region. While most of them are repeats and not very essential to the normal functioning of the cell remains perfectly healthy. Only when it is inserted into these essential regions or the gene coding regions, that is when there are certain malfunctions that develop. So the other function of these intergenic regions that is still being speculated only, it is not being conclusively proven, being speculated by researchers, is that they help in the packaging of chromosomes. They help in the proper orientation of loops and proper association of 2 different fragments of DNA, not fragments of DNA, regions of DNA that are present very far from each other. And it is considered that they play a role in genomic organization. So again, this just to put it in one slide, there is a huge DNA region that encodes for one protein, but then when it becomes an RNA, it is much more shorter. And once from an RNA, it becomes a protein, it's even shorter. Even though like a codon consists of three bases, right? Three bases of mRNA makes one amino acid. That is also a region for shorter. But apart from that, there are these regulatory elements that are present that are not present in the protein. The exons have already been spliced together. The introns have already been spliced out in an mRNA sequence because it's taken from mature RNAs only. So moving on, we have previously seen that the DNA is not just even in the mitotic phase when it is becoming condensed into chromosomes. In the interphase also, it's not just present as thread-like structures. They are also having some level of organization of the other. So one such evidence came from this experiment by this researcher known as Kornberg, and he observed for the first time that there are these beads on string-like structures that are present in the chromatin, and we can see this electron microscopy image is a really good representation of the same, that there is something that is present on the chromosome, seems like to be present in equally repetitive regions, and this entire structure later came to be known as the chromatin. And how this experiment was conducted was also very elegant. So they found that if you're digesting entire chromatin with nucleases are essentially proteins that are able to chop up your DNA. So exonucleases are those that can only chop up DNA from the ends. And when you do digest them with the chromatin, there are these fragments formed of 200 base pairs each. However, if you strip the chromatin of any associating protein and it's just the naked DNA, then there is an equal distribution of all type of sizes of DNA, and there is a smear that is observed on the electrophoretic jet. So again, this is the representation of what was observed. Say there is this DNA that is bound by proteins, then the nucleases will not be able to chop them off, because of which they are protected. This binding, this is also used to find regions where transcription factor bind, and it's typically known as a protection assay. Essentially, what it means is that DNA regions that are bound by proteins are protected from exonuclease activity, and hence that is not uh, finally observed as a smear. These regions, there will be an absence of base, and this is exactly where the protein was bound. So likewise, the is there some question? Otherwise, you can mute yourself. Okay, so to summarize, the extensive digestion of chromatin with nucleases found that there are some proteins that are constantly bound to the DNA that makes chromatin the beads on string-like structure. These particles later came to be known as the nucleosome core particles that are made up of these proteins known as histones. So it essentially contains 8 histones, dimers of H2A, H2B, H3, and H4. So they come together, and this octamer has 146 bases of DNA wrapped 1.6 times around it. It's not entirely 2 times, it's wrapped 1.6 times around it. And then there is this linker histone, H1 histone, that kind of helps the DNA bend, and it's held in place, and that lets the DNA fragment process to the binding of the next histone octamer. So this is essentially what chromatin is made up of. But again, this you would have all heard of to be the central dogma of life, and this essentially depicts that the DNA, that is our genetic material, is transcribed into RNA, and this RNA is later made into the proteins that give you all your functions. Everything that your body does is facilitated by various proteins that are present at the right place at the right time. But this requires a lot of regulatory activities, lot of proteins and transcription factors and epigenetic factors need to come to a place and at the right time. If your DNA is bound by histones and by other subunits of chromosome structure maintaining proteins, then how does it help and how do other proteins access this genetic material? So this again, the evidence first came from the identification of these group of proteins known as chromatin remodeling complexes. There are several types of chromatin remodeling complexes present in your cell. One such is depicted here. This is an ATP dependent protein, which means that it is able to bind ATP and hydrolyze ATP. So this energy that is released out from the hydrolysis of ATP is then used to alter the structure of the histone. So the nucleosome structure slightly changes when that hydrolysis happens, and that allows this protein, this chromatin remodeling complex, to slide out that fraction of DNA. So we can see here, say there is the region of DNA. Of course, there are more DNA that's present to either sides of this depiction. But say there is a chromatin remodeling complex that is associated with this particular nucleosome. Upon hydrolysis of ATP, there is this fragment goes slightly more inside and there is a sliding out of this region. So these chromatin remodeling complexes make certain region of DNA accessible to other proteins to bind to and carry out modification or carry out whatever reaction they're supposed to carry out. And this is very important. However, there is another additional question. If this chromatin remodeling complex simply binds to your nucleosome, how does it know which region of DNA it should bind to and which region of DNA it shouldn't? Because this is very well regulated, right? So if your DNA is tightly packed into nucleosomes and histones, then it will not be able to be, say, transcribed, say, made into an RNA. So in order to ensure that, your chromatin needs to know exactly which region of DNA should I make into an RNA. Otherwise, it will be a mess. All RNA are not transcribed at the same time. That is what gives your cell its a particular function. That's what makes a cell differentiated. It has a particular transcriptome, which means that it has a particular signature of RNA that it makes, and other RNAs are silenced. So this is ensured by the presence of certain sequences that we will discuss down the line. But to essentially show what other things chromatin remodeling complexes can do, this is a depiction. Again, this complex, this green thing represented over here is the chromatin remodeling complex. It hydrolyzes an ATP, and for in certain regions, what it can do is it can replace histones itself. So there are special variants of histones present. Apart from H2A, H2B, H3, and H4, there are other variants, say CNP that is present in centromeric region, or H3, other H3 variants are also present, and these give that region specificity, which we'll also see again down the line of this presentation. So a chromatin remodeling complex can do that. It can exchange these histones with other variants. And what it can also do is it can completely remove the nucleosomal complex, and this becomes a naked DNA. This is essentially what can be replicated. As we've seen, replication, transcription, translation again requires like huge number of proteins, huge number of regulatory factors that need to come together, and that needs to access the DNA, right? And temporarily it can remove these nucleosomes, and later on, again with the access of ATP, these same complexes can add histones also to the DNA to form the nucleosomal complex that is generally present. Now, again, we saw that this is the beads-on string structure, but if you look at the DNA through an electron microscope, then it is not visible to be a beads-on string structure. You have to process it a little bit. Slight dissociation needs to be done for it to take up this conformation. Otherwise, it's present as a much thicker region. This is known as the 30 nanometer fiber because the diameter of this observation, like this string-like observation that you can see under the electron microscope, has a diameter of 30 nanometers. So it's known as 30 nanometer fiber. And what it essentially has is nucleosomes much more condensed together. They are the beads on string structure is folded upon itself again, and that is what forms the 30 nanometer fiber. Now, how this is made possible is because of the presence of, again, histones only, but histones tails in particular. So if you look at this representation of the histone proteins, this is the structure that it takes up. This is the histone structure. It's known as the histone fold. And here you can see that there are multiple coil regions, but all of them, like all four variants of histones, H2A, H2B, H3, and H4, have this overhanging N-terminal tail. This is not packed into the nucleosome. Instead, it is present outside. It's present as hanging regions outside. And this allows it to interact with other tails that are hanging out from other nucleosomal complexes. So these histone tails, particularly what is found to be most essential was the tail, N-terminal tail of histone 4. That is found to be most essential for nucleosomal condensation. So what happens ultimately because of all these histone tails interacting with each other is there is a zigzag folding of the nucleosomal complex, and this gives it, as you can see, much thicker appearance, and this thick appearance is what forms the 30 nanometer fiber. But again, it is very important for the DNA to have these sharp bends in order to align into this zigzag conformation, and for that H1 histone, which we have earlier seen to be the linker histone, is very, very important. What this histone is capable of doing is it interacts with the DNA and the nucleosome, that is the other histones present, and it is able to create these bends in the path of the DNA as it exits the nucleosome. So this in between region is where H1 is present, right? And as it exists, it's able to create these bends in the DNA structure, and that is what helps arrange them into this very compact nucleosomal DNA structure. So, like we saw, there are certain regions of mRNA that are transcribed and other regions that are not transcribed, that are silenced. And this additional level of chromatin structure is what is known as heterochromatin and euchromatin. So, this is a further, an additional level of chromatin structure, that is interface of DNA, that is DNA organization in the interface phase. Here, the 30 nanometer fiber formation is restricted, and this close compact packaging is restricted to certain regions of the DNA and not the entire region of the DNA. So an example that I like to give is you need to appreciate the fact that, say, your neurons and your RBCs and your liver cells have all essentially the same DNA, and they should all have the same appearance. They should have the same mRNA, same protein present in them, and because of which they should all look alike. But that is not the case. Your neurons are much, much different, and they have these dendrites. They have this property of transmitting signals from one end to another, while your liver cells have other functions. They are able to secrete certain proteins. They are able to uh, process RBCs within them, and very distinct functions and present in the same organism containing the same DNA. This is made possible because of this level of chromatin structure regulation. It silences certain regions of the DNA and makes the other regions of the DNA more accessible, and there the gene is active, whereas the tightly packed region, known as the heterochromatin, gene is not active. This is a classification that needs to be remembered: heterochromatin and euchromatin. Euchromatin is active, heterochromatin is not active. It is more tightly packed, and euchromatin is more loosely packed. So in this image also, one such like a global view that people generally give is the presence of acetyl groups in euchromatin, the presence of methyl groups in heterochromatin. This is not entirely true, but however, euchromatins always have acetyl groups. This is appreciated, but the presence of methyl groups can be present in both heterochromatin and euchromatin. So it's not a global. You cannot just globally classify heterochromatin, euchromatin to have methyl groups and acetyl groups respectively. So the presence of acetyl groups is it has these negative charges there, so they repel each other, and because of which they are pushed away from each other, and that's what gives it the loose packaging. Whereas in heterochromatin, acetyl groups are lacking, because of which they are capable of coming to much more closer to each other. So can this also be inherited? Because that is essentially what happens. Say your liver is dividing to form more liver cells. As we know, liver can regenerate. It forms liver cells only. It does not form cells of some other region. So this chromatin structure packaging between heterochromatin and euchromatin also needs to be inherited from the mother cell to the daughter cell. How is this brought about? So the first evidence into understanding what goes on behind this came from this experiment conducted in Drosophila. Here, before understanding the experiment, we need to know that the white gene is a name that is given to the gene that is responsible for encoding the protein that gives the fly red eyes. So white gene gives the fly red eyes. If your white gene is active, the fly will have red eyes. And they found that if this is in a normal location and there is heterochromatin far away from it, then the fly has normal red eyes. But somehow some recombination happens and some inversion happens in which the daughter cell gets a gene which is present very close to the heterochromatin region. And initially, the segment that served as a barrier is now somewhere else. Then they observed that the fly does not have entirely red eyes. It has these white patches in these eyes. And this was proposed to be because of the effect of heterochromatin present closely to the white gene. They uh, proposed that the heterochromatin region is moving into the gene. The entire gene is not able to function properly, because of which there is this mosaicism seen, and the entire fly is not having a red eye. So again, this is a representation of what will happen when there are multiple genes and heterochromatin regions being recombined to form daughter cells. This was an experiment that was carried out to conclusively prove that the presence of a barrier region and the heterochromatin region is very essential in order for proper functioning of the gene. So say this is a normal fly. In this fly, there is a heterochromatin region, and then there is a barrier region which prevents heterochromatin region from spilling over into the euchromatin region. This barrier region is very, very important. And here, euchromatin is present. There are 5 genes that are encoded. Now, these 5 genes and this heterochromatin region in different daughter cells can be inherited differently after recombination. It is possible that once the barrier region is lost, the heterochromatin can first spill into, say, the first cell or the second cell. Now, in all the daughter cells of this, that is, all the clones formed from this initial embryo, it will all have heterochromatin up until the same region only. Now, there is some other recombination that happens, say more of the barrier is lost, and in this particular daughter cell, it spills on till the gene 3. Now, in this case, all the clones will have heterochromatin still gene 3. Again, say in other cells, the barrier is intact and the heterochromatin does. Can you all please mute yourself in the group unless and until you have a question? If you have a doubt, you are free to interrupt me. Otherwise, please mute yourself. So in, say, the third daughter cell, the barrier is very much intact and the heterochromatin does not spill into any of the euchromatin. Here, all the clones will have all the genes active. So again, there was this, after this experiment, it was well established that there is inheritance of these heterochromatin and euchromatin regions as well, but it was still not clear what essentially makes heterochromatin and euchromatin, and what exactly is this barrier region, and what decides what is what, like what is the deciding factor that makes a heterochromatin and silences those set of genes. This was still unknown. Later on, research revealed that there are these modifications that are present on various histones. That is what is making these modifications possible. So this is a depiction of all the different type of modification that can happen. This is also a very not updated image, because now we know that there are ubiquitination, sialation, lot of other modifications that are also possible. But this is essentially in the uh, basic representation of the modifications that are possible. So say this is the H2A, right? H2A is one. There are certain residues, certain lysines that can be acetylated. So this will be represented as H2A K5 acetylation. This would be H3K9 methylation, H3K9 acetylation. And this, that the representation that I just said now, H3K10, H3S10, H3K14, H3K23, these are all very important regions upon which methylation or acetylation plays a major role in deciding whether that particular DNA is going to be silenced or not. The H3 modifications are most commonly known in literature, whereas the other modification on other histones have also been known to be important. So different modifications on different histones means that the fate of that particular DNA is going to be different. One such example is the trimethylation of K of a lysine in histone 3. So there are few residues. Now you can see here that this residue, there's only one methylation that is represented, but there are certain residues in this. In K4 itself, there are multiple methylations possible on top of each other. So that will be Me1, Me2, Me3, and based on if it's Me2 or Me3, the fate will change. So in this case, if there is a trimethylation, Me3 present on a lysine in histone 3, then it recruits this protein known as HP1 that is able to propagate heterochromatin, which means that the spilling of heterochromatin that we earlier discussed is because of this protein known as HP1 that is recruited upon trimethylation of a residue in histone 3. So how this happens is because of a set of proteins known as chromatin readers. Now there are also chromatin writers and chromatin erasers that we will talk about. First, chromatin readers are these proteins that can specifically bind to one such modification. This is known as the histone code. So say this is lysine 4 in histone 3. This has 3 methylations. So it's a trimethylated mark. To this, there is a certain chromatin reader that is recruited. And here you can see that each of this methylation perfectly fits in its catalytic site. And if this does not happen, then this protein is not recruited, like the binding is weak and it will move away. There are different proteins for different methylation marks, different acetylation marks that are recruited in a particular region. And this can further attract other proteins, perform some other function or whatever that mark is. Meaning this is known as the histone code. That is one particular mark means it's the fate of the DNA that is wrapped around it. So these reader complexes can, like I said, can further attract other proteins that can execute the appropriate biological function. So again, this is the depiction of what exactly happens. Is say there is this DNA that is wrapped around a bunch of nucleosomes, and all these nucleosomes have these covalent modifications on its tail, different residues being acylated, methylated, ubiquitinated, whatever. And there is a specific reader that can bind to these proteins. Now, like earlier mentioned, it's not generally one protein, it's a bunch of proteins that are associated together, and that's a reader complex. So there are a bunch of histones, and this particular signature is read by this reader complex. This reader complex is first associating with this region, and further other proteins are recruited with other different catalytic activities that can bind to this region and perform the effector functions. So this can lead to a lot of things, right? It can lead to gene silencing. So if there are proteins that come, get recruited and enhance the tighter packaging of this region, then there is gene silencing. If there are proteins that are recruited that help open up these regions or like remove the nucleosome entirely, then there is gene expression. There are other biological functions as well. It can recruit histone variants, and those histone variants can give other instructions to the cell and so forth and so on. So again, here is represented three of the most important, more widely cited modifications in the histone. This is again H3K9 methylation, which means there is gene silencing. H3K27 trimethylation is again gene silencing. But H3K4 trimethylation will lead to gene expression instead. So this is why I earlier said that methylation might not necessarily always mean that the heterochromatin is formed and gene is silenced. It in some cases also indicates that the gene expression is taking place. But however, acetylation is only present in those regions where expression is taking place. So apart from reader proteins, there are also these writer proteins that are actually the histone modifying proteins, the proteins that add these modifications onto the histone. They can be acetyl transferases, they can be acetyl removers, they can be methyl transferases. They're typically known as epigenetic factors, and these different factors come together and they write onto the histone, which basically means that they add appropriate modifications to those histones, which later can be read by histone readers, and they will form whatever the function has to be formed. In this case, they have depicted how a histone reader can associate and enhance chromatin condensation in that area. So multiple proteins are associated one over the other, and this is what causes tight association of that region. However, these writer proteins are also very specific. The entire histone is not just transferred with the same kind of modification. There's a very specific region which has a specific modification. So this is also regulated based on sequences, typically sequences. One such example that we can talk about is this barrier sequence that we earlier spoke about. So here we can see that the presence of this barrier sequence prevents heterochromatin spillage. So otherwise, now like we saw, there are just reader proteins. Say there was some modification over here which was red, and now it's just spilling. All of this is getting condensed, but there needs to be a stop somewhere, right? And that is the barrier sequence over here. So what this barrier sequence does is it recruits certain proteins, specific proteins that is associated with the barrier. Here it can be, say, acetyl transferases. One such, it's easier to imagine it being acetyl transferase. So if there is heterochromatin spreading and this protein is just transferring an acetyl group, then this heterochromatin will move away, right? It will become a euchromatic region. So this barrier protein can be of many types. It can shield it can shield the entire nucleosome, or it can transfer it directly to the reader protein. It can transfer modification to the nucleosome. It can be many types of barrier sequences and barrier proteins. This is what gives the histone writers its specificity, binding to a nucleic acid sequence. There are also other situations where it is not the binding to a specific sequence, but recruitment by other proteins that can detect sequences. So there is always a bunch of proteins that is acting together, and upstream protein, once activated, will activate the downstream protein, and so on and so forth. So essentially, this can be inherited because of this mechanism only. This is the heterochromatin, and this is upon duplication. This new heterochromatin does not have any modifications yet, but what happens is this new chromatin will have these sequences that are recognized by other proteins. Barrier protein is present, does some function. There are other writers that are recruited, transfers modification. Then there are readers that are recruited, and ultimately has the same heterochromatin euchromatin structure that was seen in the parent cell. This is how it is transmitted across generations. So again, this was performed in the Xenopus embryo, and is one of the first higher organisms that were to be studied to also have this trait of transferring epigenetic modification, transferring heterochromatin chromatin structure to its progeny. Here, what they did was they enucleated the egg cell, which means that they removed the nucleus of the egg cell. This is essentially what contains the entire genetic material. So once that is removed, the cell has to have some other nucleus transferred into it that will give it its instruction. Otherwise, the cell will be just fated to die. So they transfer the nucleus of somite cells that is expressing this protein known as MyoD. MyoD is typically expressed only in muscle regions, and that nucleus is transferred into the xenopus egg. And here, after this, what happens is once a two-stage embryo forms, and then ultimately the entire embryo is formed, right? The 24 cell stage and much last to last stage, and this keeps moving on. But they found that in this stage itself, if there is no injection, there's nothing injected into this uh, uh, cell, then there is moderate MyoD expression. However, there's still MyoD expression because it was initially from a somite cell. However, if you inject normal H3.3 mRNA, which is a particular histone variant that once it is integrated into a nucleosome of a particular region, then it transfers certain traits, right? Then they saw that MyoD became highly expressed. So MyoD is a region to which this H3.3 variant of histone got incorporated and their transcription increased. So if you inject normal mRNA of this variant, then there is high MyoD expression. And if you inject a mutant of this, just as a control, then you can see there is low, even much lesser than the control cells. So this is again an example of how histone variants can help establish memory, help establish these epigenetic signatures that we call. So again, we have been discussing epigenetics. What essentially epigenetics is this: there are these changes that can be brought to the chromatin structure organization. What was a heterochromatin can be later remodeled into a euchromatin, and this is what gives the cell its differentiated trait. It's what gives it its identity. And these epigenetic changes is brought about by these reader, writer, and erasers. And this can be induced by a lot of factors. One such factor that happens normally is development. So in development, cells have different epigenetic changes. Different cells are differentiated into different cell types, and this is because epigenetic signatures in these different cells are being varied. There are also reports of certain drugs that are capable of changing epigenetics, or even in general senescence, or even diet that changes epigenetics. There are also changes from diseases, including cancer or autoimmune diseases, but also infections. Certain pathogens can come inside and alter certain genomic organization, because of which it now produces an mRNA that is required for the bacteria or the fungus, or instead of the mRNA that is required for the cell. So again, epigenetic changes is one such way by which pathogens can also take access over our cell. So moving on, in the course content, we also saw that apart from these heterochromatin, euchromatin region, there is another level of organization, and this is by the formation of loops. These are called position loops, and they are present in the chromatin region. So say this is your normal sister chromatids are present in your nucleus. There are these extended chromatin loops that are present in certain regions of the chromosome only. It's not present everywhere, and that particular region in which the loop is present is very conserved. It's in the same region across cells, and it's not randomly present anywhere. So this allows ability of far away regions to interact with each other. So we've all heard of these transacting enhancer regions, in which a region that is much far away from your gene encoding region can bend over, form specific bonds with these uh, transcription factor binding regions and enhance the transcription of a certain gene or reduce the transcription of certain genes. This transacting element is an ideal example of why you would require loops in your genetic material. How this is brought about is by the set of proteins that we've been discussing right from our mitotic sessions, mitotic phase, meiotic phase sessions, cohescence and condensance. These are SMC proteins. Basically, there are these SMC proteins that have a Walker domain, they have this coil domain, and then they have a hinge. This it typically forms heterodimers that are two different proteins coming together to form a giant ring-like structure that holds together DNA. Now, if these proteins can be used to hold together two very far away fragments of DNA, and if it's brought together in the interface region, then it will form these loop-like regions, right? That is essentially what it is. The functions that have been listed here are those from the mitotic phase or the meiotic phase, but in this case, in the interface, also cohescence and condensance play a very major role. They are used to form these loops. So how this was identified by very classic experiment. We'll be discussing 3 such experiments. So in the first place, you need to establish that there are such loops that are present, and this is because of cohesin binding. Because cohesin is one such protein that has been known to bind with the DNA and bring together two DNAs, right? In mitotic phase, it was well established that it holds together the sister chromatids before separation. So what the scientists did was they first cross-linked all the cells, genetic material, which means that in whatever interaction that it is present, it is locked there itself. It is present, it cannot move around, it cannot change. Now that interaction is then preserved. The DNA is lysed and sheared, and multiple fragments are made. Then this is subjected to immunoprecipitation by an antibody that recognizes cohesin. So whatever region is bound to cohesin will be immunoprecipitated along with the protein. This is known as CHIP, which is chromatin immunoprecipitation, which means that whatever chromatin is whatever chromatin is associating with a particular protein, that is also precipitated together with the protein. Now you reverse cross-link it, and then you purify the DNA, and then you sequence this DNA. So this was an experiment that was conducted in 1999, and here we can see that if on the y-axis they plot the relative presence of cohesin, and this is chromosome 3 of yeast, then along the chromosome, if you arrange it from 0 to 320 base pairs, then there are certain regions, say around 115, where there is a higher association of cohesin when compared to the others. These specific regions is what contains the chromatins, and this was conserved across multiple cells, and they concluded that this binding is enriched in certain regions of DNA. Now, a similar experiment, but done in a much more sophisticated manner and much more detailed manner, is sequencing of chromosome 12 in the same organism. And here they found that there are these specific regions stretching across a few base pairs long, where these cohesins could bind to when compared to the other regions. So if this is the total binding, then it is mostly located in these small stretches of sequences, and this is occurring in a consecutive manner, not necessarily in a equal interval manner, but it is present only in certain regions, and these were termed to be CARs, cohesin associated regions in the chromosome. These have been identified and well established now, but this is a paper again from 2000. And this was one of the first papers establishing the presence of these sequences to which specifically cohesin binds. Now coming to a much more recent paper, paper in 2020, they performed a similar experiment. First, they cross-linked the DNA, preserved the interaction, then they digested it with a nucleus. So only those regions that are bound by these proteins and bound to each other are preserved, and then they are ligated together. So this is known as proximity ligation, where only sequences present close to each other are ligated back. The remaining will be degraded. This is again cross-linked, it's reversed, and then it is sequenced. Now, if you plot this, on both ends there is chromosomes. So this is from 1 to say 10 base pairs, 1 to 10 base pairs. So if there is no structure, if it's a linear fragment of DNA, then the second base pair will, of course, interact with the second base pair. They are essentially the same. So there will be a linear plot that is visible on the contact map. However, if there is an interaction domain, say entirely sequenced from 2 to 10 is interacting amongst each other, then 2 to 10 region will entirely show up as a domain, the entire domain that is interacting in multiple places amongst each other. But if there is a specific presence of a loop, a position loop, where say position 20 is interacting with position 2, then there will be presence of these dot-like structures because of the interaction of these two domains. Only in this domain will there be identification of the sequence. Every other sequence would have been chopped off. So based on this, they established this graph wherein, again, this is the cohesin binding, but they overlapped it, as in you can see in the top and the left corner of the graph, they overlapped it with the presence of a position loop. So they saw that wherever these position loops are present, their cohesins are also present. So they concluded that these position loops are present only because of cohesin interaction with each other. Now, again, like we spoke about transacting elements, this is a depiction of these transacting elements. If this is your gene and this is the promoter of the gene and the factors and all have bound to it, there can be trans elements, which means elements that are present very far away from this gene that can induce, inhibit, or enhance the expression of this particular gene. These are known as transacting elements, and this requires a very accurate loops to form between regions of DNA that are present very far away from each other. And the question that was asked in this paper is, how are these position loops regulated? And they identified that cohesin is one of the most important protein that binds to specific regions that somehow it identifies, and this specific region is essentially where these position loops happen from one place to another. So moving on, we will discuss more about what they concluded when we are discussing the assignment question, previous year assignment question. So now we will move on to the previous year assignment question. There'll be 10 questions. We'll just go about solving each one of them. So the first question was, which of the following protein prevents the expansion of position loops? So again, here we have seen previously that even though the chromatin just looks like a thread of unorganized regions, it is in fact very much organized. There are these regions of highly packed domains, that is the heterochromatin. There are these loosely packed regions, which is active, genes are active and can be transcribed, and this is known as the euchromatin. There is also further association of very far away regions with each other by forming a loop, and this is the entire loop that again forms an interacting domain. But here we can see that this is essentially between these two bases. There are hundreds of bases of DNA that are formed into a loop, and these two are interacting with each other. And these interactions are what helps enhancers come together to promoters and help them carry out their function. So one such protein that plays a very crucial role in this is cohesins that we have seen previously. Cohesins bind to both these regions together, and they form this ring-like structure around these two regions and brings it together essentially in the same way that it holds together two sister chromatids. So how this was identified was by this technique known as chromosome conformation technology. This was when the birth of this technology is what enabled the scientists to carry out the experiments that are required to identify these sequences. And this is essentially what we've been discussing so far. There is a loop that is formed. There are diverse faraway fragments that are interacting with each other. And Once you cross-link them, then this association is just set. You cannot unlink them in any way. That the cell cannot unlink them. So once it has been cross-linked, you digest the chromatin. So every region that is not associated is chopped off, and the regions that are associated with each other form these small complexes that can be ligated back together. So this is again known as proximity ligation. So if these two fragments are held together in place by this position loop, then they will ligate back. The remaining will not be ligated back. Now, if you reverse cross-linking, there is this unique region that is formed that is essentially a part of the two interacting domains that were initially not together in the DNA sequence. They will be present few kilobase pairs away from each other. But because there has been a loop that is formed, every other region has been chopped off and this has been ligated back together. So these are the interacting domains that helped form the loop. So say I have an experiment in which I have to determine whether gene A and gene B, two very far away genes, are interacting with each other. Then all I need to do is cross-link, digest them, ligate them, reverse the cross-link. Now I will add primers that is specific to these two regions that I have thought of. With this primer, if there is presence of that gene, only then will this be amplified. Because one end of the primer is of one gene, the other end is of other gene. Normally, it will not be present. If there is no interaction, then these two will not be present together. There will not be a primer pair. There will be no amplification. If there is amplification, then it can be detected by running on a gel. Then I know that these two interact with each other. There are several other techniques that we will go about later. But for the sake of this question, this is sufficient. So this way, a contact map can be drawn. We will know, okay, say there is a detection over here, which means that whatever this place, this sequence corresponds to, and whatever this sequence corresponds to, both of them interact with each other. So likewise, if you arrange the chromosome in a circular manner, say this is the start and then it goes on, comes around. If you arrange it in a circular manner, there is a map that can be drawn. This region interacts with this region interacts with this, and so on and so forth. And this is known as a contact map. So in the paper that we were earlier discussing, which initially established that cohesin is responsible for the formation of position loops, what they finally conclude is there are two regions, two type of regions to which cohesins can bind. One is a very stable region that is known as the stable cohesin. They are high residency cars, cohesin associated regions. The ones that are depicted by red here, they are all the high residency cars. Now there are also these intermediate regions, the regions that do not have stable binding of cohesin. There are dynamic cohesins. So cohesin will bind and it has to be removed, and it will bind again to an adjacent region, it has to be removed, and so on and so forth until it reaches a stably bound car. Now, cohesins bound to the stable bound region can then interact with each other, form the giant ring-like complex. This will in turn bring together two of these stable cars, right, high residency cars, and that is what forms this loop at very accurate positions across different types of cells. So say here, the first and the second high residency car is interacting, third and the fourth is interacting, and then the fifth is just not able to interact with any other car. But it can also be that 2 and 3 are interacting, 4 and 5 are interacting. So this also gives it a dynamic nature. It can move from one region to another. But this is essentially how these loops are formed. This is again depiction of say there is one car and then it skips two cars, stable cars in between, and then binds the fourth directly. So this again gives a larger loop to more far away elements interact with each other and all. And within the same type of cell also, same daughter cells also, there is slight variation from each other, but in essentiality, they share these high residency cars, and together they can be plotted in the total contact map with darker regions denoting those regions that have higher possibility of forming loops, higher possibility of interacting with each other, and these lighter regions can also interact. Say fourth residency car can also interact with the first high residency car, but in a lower frequency, whereas 2 and 3 can interact with much higher confidence with each other. So this is what the paper concludes, and they also go on to identify various proteins that are required in this complex, and that is when they knock out this protein called WP1, WPL1. Now, you can see that if this is the wild type, there are these regions that are formed. These regions are the loops that are that are interacting with each other. And here we can see that upon knockdown of the WPL1 complex, there are much more of these regions formed. They are kind of extending out. Much more farther regions are also able to interact with each other, and there are many more interactions that are seen when compared to that of the wild type. In fact, if you do quantify this, the number of position loops present in the cell almost doubles when only this one protein is knocked out. So, what they conclude and what they conclusively prove is this protein, what it does, it removes cohesin away from the loosely bound region. That is, this is the protein, like I said, if cohesin can bind here also, there are dynamic cohesin, but something needs to remove it from this region so that it can go on and proceed to bind to a high residency car. This is that protein. This is what removes cohesin out from loosely bound region, because of which it can bind to a high residency car and help form exact location of position loops. If this protein is not there, that is in this case, multiple cohesins will be associated along the chromosome, and because of which any two random regions will also be able to loop together and form a position loop. So this prevents the expansion of position loops. It helps it contained to one particular location only. And that is what is the role of this protein. So to answer this question, which of the following protein prevents the expansion of position loops? It is WPL1 protein. Moving on, why was ATP gamma S used in the control experiment in the double-tethered DNA assay with the condensin molecule? So again, this was a very interesting experiment that showed that the condensin molecule bind to the DNA and requires ATP hydrolysis to form these loop-like structures. But let's look at what is the importance of proper designing of an experiment. So say there is an experiment. Your question is, what happens when I take in vitro cultured cells, any cells, say HeLa cells for this example, and you add compound A to it, an arbitrary compound A. This is your question. You're asking what happens to the cells if I just add compound A. And you observe that just addition of this compound increases the expression of a desired protein. Now, how can you experimentally show this? There is this technique known as Western blotting that many of you might have heard. So in this, what you do is, if you have in vitro cultured cells, you first pellet down the cells. Now to this pellet, you add lysis buffer. So this lysis buffer will lyse the cell membrane and all the protein will come into this soluble phase. Now to this entire protein, you run it in a SDS phase, which is again a electrophoresis only, in which it is separated based on its molecular weight. And to the molecular weight, your desired protein shows up. In that region, you can add antibodies against that protein. Now, this antibody itself can be probed, then it's known as primary tagged antibody, or another secondary antibody can be added, which can be tagged with something. So that amplifies the signal, or the signal becomes more strong and can be detected easily. So through that technique, you see that in comparison to cells that have not been treated with compound A, in cells that have been treated with compound A, there is an increased expression of one particular protein. Now, what could be a possible conclusion? Compound A enhances translation of the protein. You can conclude this. But, however, we lack any control experiment in this experimental setup. Now, how can we rule out that it is not compound A, but it's the solvent in which compound A was dissolved? Say I'm dissolving compound A in DMSO. How should I rule out the fact that DMSO is not causing this effect? This is why it is very important to set up proper controls to an experiment. Here, it can be ruled out by using something that we call as the vehicle control, wherein if you're dissolving compound A in DMSO, then there should be, along with your experiment, there should also be a setup in which to the cells you're adding just empty DMSO. In there, if you do not see an enhanced production of your desired protein, then you can say, okay, it's because of compound A, it's not because of DMSO. Similarly, the experiment that was performed was condensing binding to a DNA and forming these DNA loops. Now, how the observation that they observed was that this binding requires ATP. ATP hydrolysis is what provides the energy for this binding and formation of the loop. So you can conclude that ATP hydrolysis is required for proper binding of condensins on DNA. However, it is still possible that it is not ATP but other contaminants that is present along with ATP that brings about the change, or it is not ATP hydrolysis, it's just plain binding of ATP that is causing some conformational change that leads to this. So in order to rule this, there are proper control experiments that need to be set up. Here in this case, we can use no ATP or an ATP analog, wherein it is a special ATP that binds, that has similar structure but does not get hydrolyzed. So these are the different types of ATP analogs that are available. You can either make ATP non-hydrolyzable, make it fluorescent, or you can even tag it with an affinity label. So these analogs that are present commercially available in the market, say it is non-hydrolyzable, then what all can you do with it? It will first outcompete your normal ATP. So normal ATP will not be able to bind. So further, even though ATP binding happens, as in if it's a non-hydrolyzable ATP, it still binds to your protein that you're testing, but it cannot perform any downstream functions that come as an effect of ATP hydrolysis and release of that energy. So you can see that in your reaction, how important is ATP hydrolysis. It can also make the ATP bound state stable. Otherwise, what happens is ATP binds, it gets hydrolyzed and it's released out. ADP and the phosphate group is released out, and that state is very exists for a very short amount of time. It's not stable. So in order to make that state stable, you can use the non-hydrolyzable ATP. It can be further used to perform structural studies in which an ATP bound form has to be visualized in, say, cryo-EM or NMR, or even crystallization. You can also make ATP fluorescent. Fluorescent ATPs are also available. Here, it helps observe the environment of ATP. Where exactly is ATP going and binding? That can be visualized by a fluorescent microscopy very easily. Which region of the cell, what is happening, that can also be visualized. However, it is highly possible that the addition of a fluorescent group can alter the kinetics of ATP. So one needs to be very careful before concluding something regarding the kinetics of this ATP analog. You can also tag the ATP with an affinity label, and this can help precipitate the ATP later. So say there's a particular protein that binds ATP and you have to separate them from a mixture of protein that does not bind ATP. So you tag it with an affinity label, pull it down with that affinity tag, and then you will have this fraction of cells that bind ATP, and everything that doesn't will be separated out. So in this case, because they needed to use an ATP that does not get hydrolyzed, that is why ATP gamma S was used. This is basically a very simple modification in the gamma phosphate of ATP that is making it unhydrolyzable. So that is why it was used. It can bind the ATP binding region but cannot get hydrolyzed. You can get an essentially you can exactly view what happens downstream of ATP hydrolysis in your reaction mixture. Moving on, pick the correct statement about SMC complex. So SMC complex are these structure maintaining regions of chromosomes. These proteins we have seen previously are what cohesins and condensins fall under. So these SMC proteins essentially form a giant ring-like structure that holds together two faraway regions or two sister chromatids, or based on the state of the cell, even helps in condensation to form the chromosomes that are segregated. So the structure maintenance of chromosomes are conserved across various species. In fact, it does not even undergo mutation that easily. It is highly conserved from a small organism to a higher organism. And even single base pair changes are very rarely observed in these proteins. They are essential, of course. If they do not condense, you cannot even undergo mitosis or meiosis. So it's highly essential to the cell, and they are chromosome associated protein complexes. The other option was membrane associated, and of course, these proteins cohescence and condensance associated with the DNA. So the next question is also regarding the SMC protein only, but this time it's about what are the different domains of the protein. So what we call domains of protein is sequence. It all falls under the same sequence of protein only, but there are certain regions that have specific function. There can be an ATP binding domain, which means that is the region of protein to which ATP binds. Now, that is what is domain organization. In the case of SMC, there are these very characteristic domains. There are two Walker domains, Walker A and Walker B, on the N terminal and the C terminal. So, like we previously mentioned that 5' to 3' is the general order in which DNA sequences are listed out. For protein sequences, it's generally from the N terminal to the C terminal. So Walker A near the N terminal, Walker B towards the C terminal. And both of these are joined to this hinge region by these coiled coils. This hinge region is what is able to associate one complex with another, forming the heteroduplex orientation that we commonly see. So the answer to this would be Walker A, coiled coil, hinge, another coiled coil, and Walker B. Coiled coil are these intermediary connecting domains that form these coiled regions. And once when they're folded upon each other, they coil amongst themselves also, forming a tightly packed region that helps the interaction. So the next question was the chromosome conformation capture technique can be used to do what all? So this is essentially the same experiment, same thing that we were talking about a few questions before. Herein we will try and understand each of these different type of chromosome conformation technology so that we can appreciate what all different functions it can perform. So the first we have already discussed is interaction between known DNA. So how this happens is there is you have cross-linked, digested, proximity ligation happens, and a sequence is present. This sequence is unique to whatever interacting domains are present. Now, if you know these two domains, then you can design primers for these two domains and check. So this is done using a semi-quantitative PCR. So semi-quantitative PCR is where you run a PCR reaction, you let it amplify. So if there is presence of these two genes next to each other, then the primer will amplify and will produce this very thick band that will show up on an electrophoretic setup. So if there is band, there is interaction. If there is no band, no amplification, it means that these two genes have not interacted. These two regions of DNA have not interacted with each other. So this is only possible if you know or you have a hypothesis that these two DNA regions are interacting with each other. So this is known as a 3C technique. There are more advanced techniques that have now come up to identify even unknown sequences, interaction between unknown sequences of DNA. So there is this technique known as 4C that is based on something known as inverted PCR. So again, there is interaction between two unknown DNA. Here you do not know, say, this blue sequence of DNA. You have a hypothesis to identify whatever interacts with this red region of DNA. So how do you go about doing that? So what you do is, one is ligated, one end has this blue region, one end has this red region. So if you ligate them again together, it forms this loop, and now you use an enzyme that is present in this red region. So there is a restriction enzyme whose recognition sequence is present within the red region, and you use that and you chop it. Now you have this sequence in which both the ends are the red region. You know the red region and you can design primers accordingly. So this is a very interesting technique that was developed in order to design primers for PCR for unknown sequences of DNA. So this is what is represented here. There are two different types of PCRs that you can run. One is this inverted PCR that is really very elegant experiment. So this is a known sequence. These two are unknown sequences. So what you do is you ligate them together, and then you chop up the known sequence of DNA. So here we have this fragment wherein known sequence are the end region. So primers can be easily designed for them both. Another is by using of adapters. This is fairly straightforward. There is a sequence and you don't really know what is this particular black region. So you cannot design primers for it. So you cannot amplify that region. So in order to avoid this altogether, you add adapter regions. They are typically repeats, say 80 repeats. You attach it, so it will always attach to one end of the DNA fragment. And now you know this region, so you can design primers against this. So in this depiction, one end was known, one end was not known. So one end already we had primer. So the other end you added an adapter and you designed a primer. But you can also have situations where both ends are not known. One adapter to this region, one adapter to that region. That is brought about by very specific digestion with restriction enzyme that gives a 3' overhang. So you know, okay, this adapter, and now you add primer sequences and you amplify the entire gene. So using this is what 4C technique was carried out. So now you have these fragments that can be sequenced and you know what regions are interacting with this particular domain that you earlier thought of to identify all the interacting domains. Now there are even more advanced techniques, 5C or the high C technique. High C technique is fairly common. Many of you could have heard about it. These techniques are to get the entire visualization of how genomic DNA is organized, what loops are present where, what region is interacting with what. So this is like a omic level study. It's a global visualization. So for this, what you do is for 5C, they add universal primers. So these primers randomly attach to different regions and they amplify those fragments that can then go on and you can subject them to sequencing. Now, high C region is where, like I previously mentioned, you add adapters to both ends. So nothing is known, no sequence is known, because universal primers binds to only certain region. The coverage of this 5C technique is limited. It does not cover the entire genome. It covers most of the interacting domains, but high C covers the entire genome. It covers all the linkage between each other. So here you again precipitate reverse cross link, and then you add adapter regions to both ends. So now you add adapter, you know the sequences, you design primers, conduct a PCR, and this PCR you can give for sequencing. So essentially, we have seen that these chromosome confirmation technology can be used to identify interaction between known sequences, unknown sequences, and even identify the overall genomic DNA organization. So if you look at these options now to visualize the genome organization at a high resolution, that is possible by high C technique. Spatial relationship between known sequences of DNA is possible by 3C itself, and to yield information rich data describing genome topology. So topology is again the way in which genome is organized, and it can indeed yield information on that. So all of the above. Moving on, condensed complex participates in what? So again, condensin, coming back to the same image, condensin is one of the most important protein that in mitotic phase is able to bind these faraway regions of DNA and compact their packaging to form what is widely known as mitotic chromosome. So it helps in the chromosome condensation. It helps in chromosome segregation and cystochromatin decatenation, because again, unless and until chromosomes are formed and unless and until they are made sure that they're not wound upon each other and there are no tangles present, segregation will not be possible. There will be improper breakage, DNA breakages, and other mutations that can come up because of improper segregation of chromosomes, and condensation plays a very important role in that as well. Maintenance of genome organization, yes, we've seen that there are certain loops that can be bound by coenzyme, condense, and protein and help in genome organization. Now there is this other function, clustering of tRNA genes, that we've not spoken about yet. So if the cell has tRNA genes, they are present in various locations, like they're not present adjacent to each other. They're present in different chromosomes spread across the entire DNA. But in a cell, tRNA is synthesized in this region within the nucleus known as the nucleolus. So there is some protein that is bringing across different tRNA genes and localizing them, concentrating them into this region of the nucleus known as the nucleolus. And that protein is condensin only. Condensin only binds, again, different regions of the tRNA coding regions and pull them together and help them stay localized in one particular region. So the answer to this question would be all of the above. Condensin is one of most important proteins. In fact, knocking out of SMC proteins is often lethal to the cell. It is embryonic lethal. It cannot survive. There are certain mutations that are present in the SMC protein that also leads to very catastrophic defects, because again, imagine coenzyme protein not forming proper loops, then your entire translation, transcription of the cell is affected. mRNA protein synthesis is affected. So it is very important. Moving on, the next question was, the cut mutants in S. pombe have the following phenotype. So S. pombe is this type of yeast. It's the fission yeast that divides by basically binary fission. But what is more special about this yeast is the nuclear envelope never breaks down. So the DNA replication, segregation all happens within the nuclear envelope itself. And there is just a septum formation, a cell wall formation that chops the nucleus into two, and it's equally divided into the two daughter cells. However, cut mutants are those that lack certain essential genes, because of which what happens is, when the septum is forming and the DNA is chopped into two, then the resolution of the DNA into two daughter cells has not been completed. So what essentially happens is, there's just a bisection, there's just a cutting of the nucleus into two, but both the nucleus do not have the same content. It's not properly replicated chromatin and all. So again, cut mutants are often they stop in this stage. They do not progress further. They're often fatal. But there are certain cases in which certain cut mutants can also grow and divide, but not for long, because they have this improper segregation of chromosomes. So it has a bisected nucleus during cytokinesis, because the septum formation is only going to happen in cytokinesis. It does not happen in interphase or anything. Only when the septum is forming, that nucleus is getting bisected into two. And there are a large number of genes that have been associated with cut mutant phenotype. It's not just one mutation. It's a mutation of multiple genes having the same phenotype. So it's given one name. Interestingly, there is also observation of mutations in lipid homeostasis that is somehow affecting this process. You can read about it. It is quite interesting. So the next question was, which of the following statements is or are true regarding the SMC complex? So again, going back to this image, SMC complex, we can see, forms a ring-like structure. They have two different proteins associating with each other. Most commonly, they are a heteroduplex, and they associate with the chromatin, and they have multiple other proteins that are also present in the complex that serve various functions. So looking at the options, SMC complex have only homodimeric structure. This is of course false, because more often than not, it has a heterodimer structure present. The structure of SMC complex is X-shaped. Again, This image is a clear visualization of the fact that it is not X-shaped. In fact, it is a ring-like structure. SMC complex have ATP binding domains which catalyzes ATP hydrolysis. This is true because condensing cohesins, they all bind to this DNA based on an ATP dependent manner. ATP hydrolysis is important, and this domain, this Walker domain, has the ATPase activity. The SMC complex don't have any associated non-SMC subunits when bound to chromosome. Like we just saw, there are multiple other proteins also that are present along with these SMC complexes when bound to the chromosome. So there's only one true statement, that is option C. The next question was nucleosomes consist of what? Again, nucleosomes are this histone core wrapped around with the DNA. So it has both histones and DNA molecules. So this is a question that talks about the experiments made by Professor Koshland, and they talk about the contact map that has been plotted between the chip-seq of MCD1P. This is cohesin. This is a subunit of cohesin that they're talking about. So this map that we had earlier discussed, this is the map that they're talking about. So the options that they have given as to what are the conclusions that are made is there is only one form of cohesin complex along the chromosome that is the stable form. This is wrong because we've seen that there are certain stably bound cohesins that are bound to the high occupancy cars, and there are these other dynamic cohesins that keep binding and they are dissociated by other proteins. So this is not correct. There is a correlation between condensin complex and the looping of chromosome. There is a correlation between one of the SMC, but it is not condensin, it is cohesin complex. So again, this is also wrong. The cohesin complex doesn't slide along the chromosome. This is also wrong because cohesin complex keeps moving from one region to another, binds, dissociates. So it again slides along the chromosome. And that is how it is able to identify these like high residency cars to which it stably binds. So this is again the summary figure in which they see that there are stable cars. They are stably bound cohesin to high residency cars. There are also dynamic cohesin. Once they're stably bound, they're not removed. So these two regions will then associate with each other, forming the loop. So the answer to this question would be none of the above. So that is all that I have to discuss from my end. If you people have any questions, we can discuss that. We get the recording of this. Yeah, this has been uploaded onto YouTube. All the videos that we previously um, discussed also has been uploaded onto YouTube. The link will be shared with you soon. Then they will, NPTEL will, sorry,